Okay, this is an aqua impact presentation, and my name is Antti Kause from Luke, Finland. And feed conversion ratio, fillet yield, and disease resistance are key traits for profitability and resource efficiency in aquaculture industry. And this is because they are traits which are related to efficiency, which means the ratio of inputs to outputs. So these traits are related to reduced production costs and increased output, and also to environmental impacts like the climate change effects. And disease resistance naturally is related to fish welfare also. And in aqua impact, we develop methods, nutritional methods and breeding methods to improve these traits. But additionally, we want to demonstrate the benefit of selective breeding to farmers when we improve these traits. And this is the topic of the presentation. What is selective breeding? In traditional selective breeding, we use pedigree information, so family level information about the performance of different families. We identify high performing families and low performing families, and we select the superior individuals from the best families to be the parents, and this leads to genetic improvement. And it's well documented that growth can be genetically improved over 5 to 10% per generation using this method. But then the question is that what about the other traits, the FCR and others? And this slide shows the expected genetic improvement of FCR in rainbow trout in the Finnish national breeding program for rainbow trout. And starting from the year 94, the graph shows with the green line the FCR at commercial farms when the genetic improvement in FCR is excluded from the values. And the blue line shows the FCR when the genetic improvement by the breeding program is included into the farm performance. And the result shows that if we use the present day genetic material, then the FCR is improved by 18%. And it means directly that the feed costs are down by 18%. And we can calculate that if we assume 1 million kilogram production of fish, this means 2.6 million euros less costs for the feeds every year. And there are also positive environmental effects because FCI is a key trait for reducing the environmental impacts. These are based on the breeding program data, but we have also made a specific comparison of fish groups. And this slide is by our French partners, and they have compared two groups of rainbow trout. One group is selected for 10 generations in the breeding program, and the other group is the generation 0 fish. And they have grown the fish in a common garden experiment. And what we see is that now the FCR has improved by 19%, which is a value very similar to the Finnish data. And we also see that the fillet percentage here is improved by 2%. And now this also means that there will be 2% more income from the fillet sales. And we can again make a calculation, and if we have 1 million kilogram fish production, then and we assume certain fillet prices, then this will mean 1 to 2.2 million euros more income per year from the fillet sales. And there's an additional benefit that there will be less waste from these fish. And in both of these cases, the selection has been based on this pedigree-based selection. And additionally, for example, FCR is improved indirectly, so that we select for rapid growth, body composition, some morphological indicator traits. And this will lead to improvement in FCR and fillet percentage. A newer way to make selective breeding is called genomic selection. And in genomic selection, we can take a tissue sample from individuals, And we genotype the fish with thousands of DNA markers, and using this DNA profile, we can predict the superiority of the individual broodstock fish. And when we apply this to disease resistance, we can, for example, have a commercial farm with a large school of fish, and when they are attacked by a disease, then we will have fish which survive the disease and fish that die due to the disease. And we take a tissue sample from all the fish, we genotype the fish, and we establish the relation of the DNA profile to survival. And using this data in the broodstock, we can take tissue samples from the broodstock fish, which never encountered the disease. And using the DNA profile, we can identify the genetically best fish. So we are really looking at the individual genome of the fish, not just family structures. And this slide shows how much the selection accuracy will increase with this method. And in this case, it's genomic selection in rainbow trout to improve survival against the Flavobacterium columnare disease. It's a major disease in the fingerling phase in rainbow trout, and actually other species too. And on the graph here on the left, on y-axis, we have selection accuracy. And this means the efficiency or the accuracy at which a breeder can predict the genetic superiority of the fish to survive or to die under this disease. And looking at the x-axis, we see that if we use some hundreds of DNA markers, this is a typical pedigree type of breeding. So we more or less establish the family relationships and family pedigree. The accuracy is low, but when we use thousands of DNA markers across the genome, this is genomic selection, and the accuracy is increased, the selection accuracy. And the increase in the accuracy is directly translated into a more faster genetic improvement when we improve this trait. So this proves the efficiency of genomic selection. And on the graph here on the right, we have for broodstock fish, which are alive, the expected survival of their offspring when they encounter this disease. And we can see from the graph that there are broodstock fish which for their offspring, we expect that their survival is 94%. And there are broodstock fish, those offspring will likely have survival of 34%. So there's great variation in the broodstock, and we have been able to estimate it through the DNA profile of the fish. And naturally, we select the parents with the highest expected survival in the offspring. So this is genomic selection. Okay, the current status, all these results come from ongoing breeding programs from which the fish are distributed to the farmers. So this is already implemented and in use, and of course the breeding companies benefit, but the impact goes through farmers, processing industry, and to the environmental impact. The genomic selection itself has been implemented in a commercial breeding programs starting from 2016. And in Aqua Impact, we develop more sophisticated ways for genomic selection, and especially more cost-efficient ways to implement it, because there is great interest to implement it in more and more breeding programs, and this is our focus. Thank you. The contact information is here, and please follow us in Twitter, and in the Madeiran Congress we will have 11 contributions, and we hope to see you there also. Thank you.